So this is the last week of class, right? So we've got 2 lectures. In lab is a review day. You'll have your lab exam on Thursday. The lab exam covers exercises 11, 12, and 13. If you have not had lab yet, there's a bunch of practice tests for you to do in lab to kind of review for that. What we're going to do for this week is we're going to cover special senses, and we're going to primarily focus on how your ears work and how your eyes work. And so the physiology is fairly straightforward. I don't think it'll take a lot to master that information. So we've got 2 lectures to cover special senses. There will be a review next Tuesday, starting about 10 o'clock and 2:56. I will post this on Blackboard, but if you want to review for the final exam, we will have a review session scheduled for next Tuesday at 10. I have handed out your exam. I will post a key up. People did rather well on this test. There was about a 75 average in the class, so it was a decent average. So you probably should start reviewing. If you have not done so, start reviewing for the final exam now. So for the final exam, it's going to cover articulations. We talked about synovial, we've talked about fibrous, cartilaginous, different types of joints. We've talked about how you classify them based on function. They're synarthrotic, amphiarthrotic, diarthrotic, how much they move, right? So you want to start looking at those: flexion, extension, adduction, abduction. You start reviewing muscle physiology. So we covered muscles. And then the nervous system. So we talked about different types of cells that you find in the nervous system: oligodendrocytes, astrocytes, right? And what those do, as well as how a neuron establishes the resting membrane potential, right? Passive channels, always open, potassium, sodium, potassium, ATPase. Okay? So those are the things you want to start reviewing, and then special senses will also be on the final. So those are the topics that we will cover. Okay? Do you have any questions so far? The final exam will be structured exactly as every other exam that you have taken in here. So there will be 35 multiple choice questions, there will be labeling of diagrams, and some short answers. Okay? So it will not be 2 hours. Take it 2 hours. All right, you guys okay so far? All right, so let's get started on special senses. I've got 2 slides on other senses, just to kind of, cover the bases. So olfaction, right? That's your sense of smell, right? The nose, right? The stimulus for olfaction is chemical, right? So you remember how we talked about different channels and how you could open different types of channels? We had chemically regulated channels, we had voltage regulated channels, we had mechanically regulated channels. Olfaction uses chemically regulated channels. When receptors in your nose bind to a chemical, it will change how a channel functions. It will open or close it. Now, the part of your brain that interprets smell is called the uncus, right? And this is located in the temporal lobe, the medial part of the temporal lobe. So that's the part of the brain where the signals are sent. And this system adapts very rapidly. Do you remember what adaptation is?
Exposed to stimulus for a long time.
Yeah, you stop the action potentials, right? And we said that if the stimulus is constant, the frequency of action potentials in the presence of a constant stimulus will decrease over time. So this system adapts very, very rapidly. You can smell an infinite variety of qualities because the way you interpret smell, it's not that a receptor is bound or unbound. It's what type of receptors are bound. So you have a number of different types of receptors, and it's the overall pattern. Okay? So that gives you the ability to distinguish small differences in smell, right? Because you're looking at the overall pattern of receptor activity. So where are your receptors going to be located? In your nose, right? Which part of your nose? Superior nasal cavity, up high, right? Receptors will bind, that's going to activate the neurons. Neurons will send action potentials. Where do the action potentials travel? What cranial nerve? Olfactory, right? Cranial nerve 1, remember the olfactory bulb, olfactory tract. That information then will be sent to the temporal lobe, to the uncus, right? The only other thing I want to add is that sometimes people suffer from seizures, right? And these are called uncinate fits. So a seizure is the spread of inappropriate electrical activity, right? So we've got inappropriate electrical activity in the uncus. How will that be perceived? What's that going to cause? What? You smell things that aren't there, right? You're going to hallucinate smells, okay? Because you're affecting the part of the brain that interprets smells, okay? So you'll actually hallucinate or perceive smells that are not there, okay? All right, any questions on olfaction? Okay? Gustation. Gustation is your sense of taste, right? Gustation is sense of taste. The stimulus is going to be chemical, right? Our receptors for sense of taste are chemicals. Where do those receptors reside? Tongue, right? What particular part of the tongue? Taste buds, right? So our circumambulate papilla has chemical receptors, right? And you actually have different types of receptors that detect different qualities. So there are receptors in your tongue that detect a sour taste. When you're talking sour, what you are measuring is pH. If something has an acidic pH, it will taste sour. Okay? So that is reflected in the pH, right? And remember that was concentration of protons. Right? Concentration of protons. Sweet is typically you are binding to organic types of compounds. Right? So carbon, hydrogen, oxygen, organic compounds. Okay. Salt is going to be any type of ion that is not a proton, right? Because protons, we said, taste sour. Salt, right? Sodium chloride, potassium chloride, that makes food taste salty, right? And you'll note that those do not ionize to make protons, right? The last taste quality is bitter, and these are things like quinine. You are actually most sensitive to bitter flavors, right? You don't need as many receptors to detect that quality. Why do you think that is? What in life is bitter? Right? Poisons, toxins, things that will kill you are bitter flavors. Okay? And so that you have developed this kind of evolutionary sensibility to be very sensitive to bitter flavors because toxins and poisons are bitter tasting, and so you can detect them at a low level. Number of different cranial nerves actually monitor taste buds. So we've got glossopharyngeal, and the vagus nerve monitor those taste buds. And we're going to send the outputs to the primary somatic sensory cortex. Is this pre or post central gyrus?
Post.
Post central gyrus, right, is your sensory cortex. So then you remember the sensory homunculus and a picture of the face with the tongue, right? So that's where that information would be sent, that part of the sensory cortex. And again, much of what we perceive about our sense of taste is actually olfaction. When you have a cold, things taste different, and it's not because your taste has been modified, it's because your sense of smell has been modified. All right, that's it for gustation. You have any questions about sense of taste? All right, now we're into kind of the real meat of the special senses. Audition, which is your sense of hearing, and equilibrium, your sense of balance, are all handled by the ear. And so we're going to focus on the ear and talk about how we actually perceive sound and perceive equilibrium, which is basically where your head is in three-dimensional space. Now, we've talked a lot about ear structure because we've done this in lab, so this is kind of a review. What I want to point out is that we now have the ear divided up into parts, right? We've got our external ear, our middle ear, and our inner ear. And you want to note the differences in those and whether they're air-filled or fluid-filled, all right? So we said external ear, we've got the auricle, right? Big funnel outside the head catching sound waves. We've got the external auditory meatus, right? We've seen that before. That's the big tunnel that runs through the skull and the temporal bone, right? And then we've got the tympanic membrane. So from here out, that is considered external ear. Is that going to be air or fluid filled? Air filled, right? Swimmer's ear, so when you get an infection out here, right? And you get fluid building up in that external ear, right? Tympanic membrane, we start into our middle ear. We have our auditory ossicles. Do you remember the names of those? Malleus, incus, stapes, right? And we said that one end rests up against the tympanic membrane, and the other end sits on the oval window, another membrane, right? Now, is that middle ear fluid-filled or air-filled? Air-filled, right? So that's all air-filled as well, all right? Then we hit the inner ear. And the inner ear, we have several structures that do different things. We have the vestibule, right? And you remember that is what deals with equilibrium, acceleration, deceleration, and rotational motion, right? So that's what detects or helps you perceive, you know, what position your head is in space and whether it's moving or not. Then we've got the cochlea, right? And that looked like a big snail. And that was where our hearing apparatus was located. We also have our semicircular canals. Again, rotational motion is detected there. Now, these are all part of the inner ear, our semicircular canals, this vestibule region, the cochlea, right? Is this going to be air-filled or fluid-filled? Fluid-filled, right? So these are all fluid-filled structures. Then we've got cranial nerve 8, vestibulocochlear nerve. And it's got a branch, right? One branch goes to the cochlea, one branch goes to the vestibule semicircular canal. So, what we need to do is talk about kind of the steps in hearing sound, right? Sound waves, right? We talked about, and basically sound waves are simply pressure changes in the air. So we're going to get sound waves. And we're looking at time versus pressure. So that's what sound waves are, right? Changes in pressure, right? And that will actually, when that travels into your auricle, down your down your external auditory meatus to the tympanic membrane, it will actually cause the tympanic membrane to move, right? Now that tympanic membrane is not going to move much, okay? So the tympanic membrane will move about 1 micron, all right? And that's 1 times 10 to the minus 6 meters. 1 times 10 to the minus 3 meters is 1 millimeter. And that is the thickness of a penny. Okay? So if you took and sliced a penny into 1,000 pieces and took one of those slices, are you with me so far? Take the thickness of a penny, take it, slice it in 1,000 pieces. One of those slices is how much that tympanic membrane will move back and forth so the sound hits it. All right? It's not impressive. Don't be impressed, right? That amount of movement, that's just very little, right? So we're not moving that tympanic membrane very much, okay? So what we've done is we've taken these pressure changes and we have produced mechanical movement, right? Because our tympanic membrane is now moving. So we've converted pressure waves into motion. Moving so far? Pressure waves are now motion. Now, you remember all of our ossicles sit up against the tympanic membrane, right? Our malleus, incus, and stapes, right? Those are going to conduct those vibrations to the inner ear. Our ossicles were in our middle ear. Middle ear is air-filled, right? We're now going to take those vibrations to the inner ear. What's the connection between our ossicles and the inner ear? What membrane? Oval window. Right, so that stapes sits up against the oval window. All right, so let's take a look at the inner ear, right? The inner ear is fluid filled, right? It is normal to have fluid in that region. It's filled with a fluid called endolymph, right? And hopefully you remember from lab, we talked about how membranes are going to move in the inner ear, right? And those membranes are going to be fragile. If you can move membranes with a liquid wave, right? So we're taking liquid waves, we need to put them into something that's going to protect them. And so the outside of the inner ear is called the bony labyrinth, right? And this protects a membranous labyrinth on the inside, okay? Now, our inner ear, we divided up into the vestibule, the semicircular canals, and the cochlea, right? Vestibule was our utricle and saccule. We'll cover that in just a little bit. Semicircular canals were the big loops. We said those detect rotational movement. And then the cochlea is where you have your hearing apparatus. Okay? So, vestibule, we've got sensations of gravity and linear acceleration. Semicircular canals contain our ducts, which are membranous, and these detect rotation, right? Cochlea contains the cochlear duct. And we looked at a histology slide of that. We kind of had two little membranes sitting on top of one another. And we'll talk about that a little bit. But the cochlear duct is where you have receptors that give you your ability to hear sound. All right, so just to kind of review, our outer ear and middle ear were air-filled, right? Our inner ear was fluid-filled. Okay? So what sits between the outer and middle ear? What structure? That's where you get the battery. What membrane? Tympanic membrane, right? So we've got the tympanic membrane sitting between the outer and the middle ear. The auditory tube equalizes pressure between the outer and middle ear, okay? So you've got an auditory tube that runs from the middle ear into the oral and nasal cavity, right? And it is used to equalize pressure between the outer and middle ear. What can happen if you do not equalize pressure between the outer and middle ear if there's a large difference? Pain, and you can rupture the tympanic membrane, so you blow out your eardrum. Okay? So that's important to keep the pressure on either side of the tympanic membrane equal. Right? Otitis media, otitis, ear, Media, what part of the ear are we looking at? Middle, right? So this is a middle ear infection, right? Get inflammation of the middle ear. When you get inflammation, you usually get fluid buildup, right? Are you supposed to have fluid in the middle ear? No. What's that going to do to the tympanic membrane? If you have fluid building up in the middle ear. So it's pushing on the tympanic membrane, you get pain. When you have kids that have middle ear infections, what do they do to their ears? Rub them, right? Because it's painful, they sit there and just pull on them, right? Okay. So here is a picture of the inner ear. We said the inner ear is fluid filled, right? The blue is our endolymph, right, a fluid. We've got our bony labyrinth, this part that surrounds the membranes. Here is our semicircular canal. Here is the vestibule. Here is our cochlea, which contains 3 different ducts. And we're going to take a look at these ducts in detail. The cochlear duct is the important one because it contains your hearing apparatus. Okay? All right, so you've seen a picture like this in lab. Okay? Here are these sections that are basically, we talked about how you take in Play-Doh and make Play-Doh worms, and then you can roll them up and make them look like a snail, right? That is basically how your cochlea is constructed, right? This is all part of the same Play-Doh worm, right? We just coiled it up and sectioned through it, right? And we have 3 different ducts, right? We're going to have the cochlear duct, which contains our hearing apparatus. We've got our vestibular duct and our tympanic duct. So we've got our vestibular and tympanic duct here, and then our cochlear duct that sits in between them, right? All fluid filled. Okay. Now, your hearing apparatus is found in the cochlear duct, okay? This is called the spiral organ or the organ of Corky. This is your hearing apparatus. This is why you hear things, okay? Now, the way that this works, right, is that we've got a membrane on the bottom and a membrane on the top found in our spiral organ, okay? The membrane on the bottom has hair cells sitting on it. And these hair cells, right, have cilia that extend up and point towards a membrane that sits on top, and this is called the tectoral membrane, right? So the basilar membrane is the bottom, B for bottom, tectoral is the top, T for top, okay? And what happens is that our oval window is going to move back and forth, right? What's pushing on the oval window, making it move back and forth? Stapes, right? We moved our ossicles vibrated. We now have our oval window, right, moving back and forth. And our oval window right, is going to separate our middle and inner ear. Inner ear is fluid filled. Okay? Our oval window is going to vibrate. It's going to move back and forth. It moves back and forth about 22 microns. Compare that to how much our tympanic membrane moves back and forth. How much did our tympanic membrane move back and forth? 1 micron. So how much have we amplified the signal? 22 times. We made a much bigger change in our oval window compared to our tympanic membrane, right? So this is where we have amplified the signal, right? We've got a bigger, much more movement, okay? Now, that oval window is going to move back and forth. What is on the non-ossicle side of the oval window? Okay. Fluid, right? We said inner ear is all fluid filled, right? So what's going to happen to that fluid? It's going to move, right? So we're going to create waves, right? And the waves are going to travel through those tubes. right? And we have the vestibular duct, the tympanic duct, right? What happens is when those fluid waves travel, that basilar membrane is going to go up and down. All right, so our basilar membrane is going to go up and down. What's sitting on top of our basilar membrane? What sits above the basilar membrane? Our little hair cells. Okay? What's above the hair cells? Tympanic. Right? T is for top. So if we've got this membrane going up and down, we've got hair cells sitting on top of it, and we've got a roof to that. We've got the tympanic membrane sitting above the hair cells. What's going to happen to the hair cells in the tympanic membrane? They're going to collide, right? When they collide, those hair cells are going to bend. What type of receptors are we going to have on our hair cells? Mechanical, right? We're going to have mechanical receptors, right, that are going to open, or mechanical channels that are going to open up when we bend those hair cells, right? Now, do you see this? What's been yellow on all of our models? Nerves, right? So we're going to have branches of our cranial nerve 8 innervating these hair cells. You're going to open up channels by activating the mechanical receptors, and we're going to get action potentials traveling down branches of cranial nerve 8. Is it making sense so far? All right, so this is just a larger magnification. Hair cells. Which membrane moves due to sound? Tectorial membrane. I read that wrong. Tympanic membrane, sorry. Right. Which membrane is going to move? Is it going to be the basilar membrane or the tectorial membrane? Basilar, the bottom. Right, basilar membrane with the hair cells is going to move back and forth, and it's going to hit that tectorial membrane. All right, so let's look at all the steps now. We've got sound waves. Here are our pressure differences in air. Sound waves traveling down our external auditory meatus, and they're going to hit that tympanic membrane. Tympanic membrane is going to vibrate. When it vibrates, it's going to cause all of the ossicles to vibrate. When all the ossicles vibrate, that oval window will start creating waves in the fluid in our inner ear. The pressure waves are going to distort the basilar membrane, right? They're going to make that basilar membrane go up and down. The vibrations of the basilar membrane causes the hair cells to press against the tectorial membrane, right? That is going to give you information about sound. So we've got to talk about a couple things about sound intensity and pitch, but that information is going to be relayed down the cochlear branch of cranial nerve 8. You guys okay with how the ear works so far? So, we've got two different things about sound that you need to be able to figure out. Is it a high-pitched noise? Is it a low-pitched noise? And how loud is it? Right? So we've got to be able to code that information in the action potentials that are traveling to the tympanic, the temporal lobe of our brain. Right? So, a couple different things about sound. Right? One is the pitch. And we're going to use frequency. It's not exactly the same, but it's pretty close. So we're going to use the frequency to show us pitch. And frequency is simply the number of waves going past a point over a certain period of time. pressure. Okay. So frequency, right? We're going to say reflects pitch. Is it a high pitch or a low pitch? Right? Here, if we look at 1 millisecond, we have 7 waves. Here, if we look at the same time, we have 1 wave. Okay. This is going to be low pitch. This is going to be a high pitch sound. You guys okay? Right? So these are sound waves coming in. Here we've got a lot of waves coming in over a short period of time. Few waves, longer period of time. Low pitch, high pitch. That's simply the sound waves coming into your ear. Okay? Now, humans can hear, the frequency is measured in hertz, and that's simply cycles per second, waves per second. Okay? Humans can hear between 20 and 20,000 hertz. Right? That's the frequency we can hear. Can dogs hear outside that frequency? Yes. Dogs have a much greater ability to hear higher pitched sounds than we can hear. Okay? So there are other animals that can hear much better outside of this normal range. Okay? Next, we'll talk about how loud the sound is, right? Now, you notice when I drew this, I did my best to keep the amplitude or the height of the wave the same, right? So this would be the same intensity sound as this. Neither one of those are louder than the other, right? The intensity, how loud, is the pressure change, right? So, so if I drew it like this, right? Which is going to be the louder sound, red or purple? Red, right? Because it has a higher amplitude. The pressure difference is greater, right? Would it be a different pitch? No, right? Because I drew the same number of waves, right, over time. Okay? So we've got two different things. We've got the height of the wave. That's how loud it is, right? The greater the pressure change, the louder the sound, right? We measure this in what's sound pressure units called decibels, right? The threshold for human audition is 0 decibels. Right? Alexander Graham Bell is where you get decibels from, right? Normal conversation is 50 decibels. I'm talking slightly louder than 50 decibels. But I am nowhere near your pain threshold, right? Pain threshold is 140 decibels, right? And that's when you have something so loud that it hurts your ears, okay? All right. So, what's the units when you're dealing with pitch? Hertz. Dealing with the intensity or loudness of sound? Decibels. Okay? So we've got to be able to code for both of these, right? Because you can tell if you're listening to a loud sound or a high-pitched sound or both, a loud and high-pitched sound. So we have to be able to code for that with our apparatus that we're working with, okay? Now, do you remember when we were talking about sensory information, pressure, how do you know when you have an elephant standing on your foot versus a chihuahua, right? What coded for that? For like pressure, pain, what coded for the intensity of pressure, the intensity of pain? Frequency of action potentials, right? That is exactly the same for the ear. If you are listening to a loud sound, the frequency of action potentials is going to be high. Okay? Now, some of you are starting to look at me a little weird because, right, when we were talking about pitch, we were talking about frequency, right? But we were talking about the frequency of sound waves, not action potentials. So you want to separate those, make sure you have those separate, okay? So when you're talking how loud it is, right, you are talking about increasing the number of action potentials traveling down cranial nerve 8. You guys okay with that? Distinguish between the frequency of sound waves, right, pressure changes, and the frequency of action signals. Totally different things. You guys okay? All right. Now, pitch, right? You want to think about pitch more like the sensory homunculus. Okay? What's the sensory homunculus? Kind of a map of the body, kind of wrapped around the sensory cortex. Here what we're going to have is we're going to have our auditory cortex, which is part of the temporal lobe, and that should make sense for you too because your ear sits right on top of that temporal lobe. And can you see how it's been color-coded in this diagram? This purple end, low frequency sounds. This yellow end, high pitch, high frequency sounds. Right? So if I stimulate this yellow end of the temporal lobe, what are you going to hear? High-pitched sound. If I stimulate the other end, low-pitched sound. Okay? Now, you'd be saying, okay, well, how do you know which end to send it to, right? Our cochlea is, we said it's one long Play-Doh snake, Play-Doh worm, right? When we coiled it up, we're taking this, we're going to make that the center. Right? We said the basilar membrane will be displaced when you have those liquid waves traveling through. Right? The basilar membrane at the center end, down here, will be displaced by low-pitched sound. The basilar membrane down here towards the end over here is going to be displaced by high-frequency sound, high-pitched sounds. And that's how you know where to send it in our temporal lobe. You guys okay with that?
Wait, why do people respond typically better to low pitch sound, or like lower voices, lower than they do high?
Right, you start, as you age, you start losing that high pitch end. It's that you cannot hear the high pitch end.
Okay.
You start losing that. Okay? All right, so if I ask you how you code for intensity of sound, intensity, how loud it is. Frequency of action potentials we're firing, right? Well, what if it is a very loud, high-pitched sound? It's going to go towards the medial side, right, towards of the temporal lobe, right, that auditory cortex. You guys okay with those two? Okay. All right. So the only other thing I want to kind of show you is we've got this nerve. It travels into our brainstem and goes up to our inferior colliculi. Why is sound traveling to the inferior colliculi? What is the function of the inferior colliculi? Superior colliculi, reflex response to visual. We had inferior colliculi, reflex response to auditory. So why do we have to send information there? You're going to generate a reflex response. You have to get the signal, right? So travels right up inferior colliculi. It's going to go to the thalamus. What's the function of the thalamus? Big filter, sensory filter, thalamus to the auditory cortex. Okay? Now, this is bilateral. So if you hear in your left ear, both sides of the brain are going to get that information. Okay, so it is a bilateral. It will go to both sides. Okay, so let's go over and talk about the vestibular portion of the ear. Vestibular portion is your sense of equilibrium. Where is your head in space? Okay? We've got utricle, saccule, and semicircular canals, all fluid-filled. right? Well, still inner ear, all of them are fluid filled. We've got a bony part surrounding it, bony labyrinth, covering up the membranous labyrinth, all the membranes on the inside. Okay. Utricle saccule provide information about acceleration and deceleration, right? And we talked, I think, about this in lab, where you've got a shoebox full of rocks, and you're driving in a car, you slam your foot on the brake, rocks slide forward, you slam your foot on the gas, accelerate. That is exactly how these work. You have what are called otoliths. These are ear stones, right? And they sit above hair cells. So when those otoliths slide back and forth, they will bend these hair cells. Okay, does that make sense, everybody? All right, so here we've got our otoliths, right, our ear stones. We've got our hair cells, right? When you accelerate, the otoliths will slide one direction and bend those hair cells. When you decelerate, stop, the otoliths will slide the other direction, bending those hair cells. So one direction will turn on action potentials, the other direction will turn them off, right? On-off switch. And that's simply because these otoliths lag behind when you move. Okay. What type of receptors are going to be on our hair cells? Mechanical, right? You're going to activate them with motion, movement of those hair cells. Right? Now, you remember what the semicircular canals look like? How many loops? Three, right? 3 different planes, 3 different ways to rotate, right? You have hair cells that extend into that canal, right? And we said that's all fluid filled, so you're going to have waves rotating through there. What happens when a wave comes through there? Hair cells will bend, okay? So we've got these loops, all 3 planes. What type of receptors are going to be on our hair cells? Mechanical receptors on our hair cells. When they bend, you're going to activate these. We've got sensory nerves monitoring those hair cells. Okay? So here you can see, right, direction of rotation, fluid through the structure. Hair cells being bent as you move. We've got a number of different, again, you have the number of different branches of cranial nerve 8 monitoring those hair cells. They're going to send information down cranial nerve 8. Which branch of cranial nerve 8 are you going to utilize? What's the name of cranial nerve 8? Vestibular branch, right? It's the vestibular cochlear nerve. You're going to use the vestibular branch for that information, right? All right. Has anyone ever had Meniere's disease? No. Okay. Meniere's disease is a type of kind of an inflammation of the inner ear. Tell me the 2 functions of the inner ear. Balance, rotation, and what happens in the cochlea? Sound waves, right? Interpretation of sound, okay? So if we get a type of inflammation of that inner ear, what do you think Meniere's causes? Balance, vertigo, and hearing things, right? You get tinnitus, which is ringing in the ears, right? Ringing in the ears and vertigo, because you were disrupting the function of those 2 parts of the inner ear. Does that make sense, everybody? Right. And vertigo, I don't know if any of you have ever had vertigo. This is absolutely paralyzing. Right? The room is spinning, and you're not. And it is just terrible. You can't drive, you can't walk across the room. It's really, really horrible. All right. Are you guys okay with the ear? You can do linear acceleration and deceleration, explain how that works. You can explain how detection of rotation of the head works. And you can explain how you hear, how the ear functions to hear sounds. Sure. Yes, we've got it. And it's pretty straightforward compared to some of the other stuff we've covered. This is not bad. You got to say, this is not bad. You guys okay? I'm getting ugly looks. Good? You guys okay? Okay. So what I'm going to do is very briefly start talking about vision. We won't get through all of it, but I want to start it, and then what we're going to do is we're going to take a break, and I passed out a handout that has questions about ears and eyes. And so we're going to go and start doing some of those questions. I'll pick out which ones we've covered so far and you should be able to answer. All right, so the eye deals with detection of light, right? And so we talked a lot about this in lab, right? We've got 3 different layers of the eye. We've got the cornea and the sclera, which are all one layer. Beneath that layer, we have the choroid layer, which is the dark pigmented layer. And then beneath that layer, we have the retina. And the retina is where you have photoreceptors, receptors that detect light. Okay? We have to get light into the eye, right? So the cornea and the lens are clear, and light will travel through it. And when it does travel through the cornea and the lens, it gets bent. Okay? Have you ever taken your arm and put it in a bathtub or a swimming pool, and it looks like your arm is bent where it goes into the water? And it's not. You can try it if you don't want to put your arm in, you can try it with a straw. Go fill your sink up with water and put a straw into the water. It will look like the straw is actually bent, right? That's because when light travels through a new medium, a new material, it can bend. And that's exactly what the cornea and the lens do, is that light will come in and it's going to get bent. Light rays will come in, light travels in a straight line until it hits a new medium, and then it will bend. So the cornea and the lens are what are called refractive. They will bend light. Okay? Now, we can control how much light comes into the eye through the size of the pupil. Right? The pupil is the dark hole. When you look at yourself in the mirror, you have a dark dot in the center of your eye, and that will actually change size. The iris, which is the colored part of the eye here, controls the size of the pupil. Okay? So when your iris gets really big, your pupil gets small. When your iris shape shrinks in size, that pupil is getting bigger. It's very much like an accordion, right? So your accordion spins out, your pupil is getting smaller. You squeeze the accordion gets smaller, your pupil gets bigger, right? So that's how your iris works. It actually folds back on itself to increase the size of the pupil. Right? We've talked about the conjunctiva. That's an epithelial layer that covers the cornea and the sclera. It can become inflamed, and when you get conjunctivitis or pink eye. We've got fluid-filled areas here and here, anterior chamber, posterior chamber. We'll talk about the fluid that fills that and why that's important. And then we've got the posterior cavity, and that was filled with a big jello ball, like you saw in lab, the vitreous body, or vitreous humor. All right, so let's talk about the pupil first, right? We've got 2 different types of muscles that are responsible for controlling pupil size. We've got these radial muscles, these circular muscles and the radial muscles, right? Radial muscles will be responsible for dilating the pupil. These circular muscles will be responsible for constricting the pupil. So you remember how we talked about parasympathetic and sympathetic effects on the eyes, right? The muscles that cause dilation of the eyes, that's sympathetic innervation, right? You remember sympathetic activation causes you to collect more visual information so you can see where to escape from the shark that's chasing you, right? Then we have the parasympathetic will actually innervate these circular muscles, right, that cause the pupil to constrict, right? Because this is after you've eaten your turkey and you just want to curl up and sleep on the couch, right? Your pupils actually get smaller. All right. Now, we also have a structure called a ciliary body. Take that out. Here. Ciliary body is this and this, right? You've got ligaments that link to the lens from the ciliary body. So the ciliary body has a couple of different really important functions, right? It will extend to the level of the ora serrata, right, where you don't have any receptors located on this part of the eye. It contains ciliary muscles that have suspensory ligaments that attach to the lens. So what that combination of the muscle and the suspensory ligaments do is they actually can change the shape of the lens as what you are looking at gets closer or further away, right? So if you're looking at something, so I'm now looking at the periodic table, right? My lens has to actually have a different curvature than if I'm looking at a piece of paper right in front of me, right? That is what this body does. It will actually adjust the curvature of the lens for distant vision versus nearsighted vision. Okay? And we'll talk about that a little bit later. But this muscle and these suspensory ligaments, the lens doesn't curve quite well as you age, right? So this process doesn't work well as you get older, and that's why when you get older, you end up with reading glasses, because you cannot adjust to nearsighted vision, because this process isn't working well. All right, ciliary body, the other important thing it does is it actually makes aqueous humor. And aqueous humor fills the anterior and posterior chambers. Remember the chamber that sits in front of the iris and that little tiny chamber that sits behind the iris are filled with aqueous humor. That is made by the ciliary body. Right? So we're going to produce aqueous humor by the ciliary body. Right? So we've got chambers of the eye. Right? We've got that large posterior cavity, our vitreous chamber, our vitreous body sits back there. And then our anterior cavity has our anterior and posterior chambers. All of this, again, filled with aqueous humor produced by the ciliary body. Now, the reason that that is important, right, is that the aqueous humor is going to circulate within the eye, and it will actually drain out of a canal. This is called the canal or Schlemm. It goes back into the bloodstream, right, because if you produce a fluid, you have to drain it, right? Now, the deal with the canal of Schlemm, do you remember what I said the iris was like? What analogy did I give you for the iris? Functions like an accordion, right? Functions like it folds back on itself. When you dilate the pupil, the iris folds back, and it will cover the canal or Schlemm. Okay? What happens when we cover the canal or Schlemm? What happens to our aqueous humor? Can't drain. It's going to stay in the eye. Can't drain, right? When you have too much fluid in the eye, what happens to pressure in the anterior chamber? It's going to go up. And that creates something called glaucoma, right? If you have elevated interocular pressure, if you have too much aqueous humor in that anterior chamber, it's going to create a clinical condition called glaucoma. Now, when we look at the sclera, that sclera is not flexible. It does not blow up like a water balloon, right? It stays the same shape, right? What is the only exit out of the back of the sclera? Posterior. What exits out of the posterior side of the sclera? Optic disc, right? You've got the optic nerve and the retinal arteries, the retinal veins, right? So if we've got lots of pressure, where's the weak point in the eye? Optic disc, right? So what happens if you squish that? Start compressing that cranial nerve too, that optic nerve. Okay? So that's why this is problematic, right? Glaucoma is very bad to have if you have this increased interocular pressure. It will actually the weak spot is the optic disc. It can push that optic nerve out. It can start damaging that. All right? So, if someone has glaucoma, would you want to give them a sympathetic drug in the eye? Why not? What is sympathetic? Dilate the eyes, and what happens? Block the canal. And that's going to make interocular pressure go even higher. So what they always do in the order, right, they always check interocular pressure first before they dilate your eyes at the eye doctor. If you ever go in, they're always going to either blow air into your eyes with puffer, or they will take and numb your eyes and put a little suction cup on it, and it will sit there and click until you get the right reading, right? They'll always do that first to measure this, and then they give you the sympathomimetic drug to cause the dilation of the eyes because they are okay to block the canal if they know your interocular pressure is normal. Does that make sense, everybody? Why you do it in that order? It's always done in that order. Okay? All right. Okay, so we've got the 3 layers. Sclera with the white, choroid is the pigmented, retina has the photoreceptors, has the light receptors. Right? So, what we need to do is talk a little bit about light coming into the eye. So, if you look directly in front of you, whatever's your center vision, right, that will travel through the cornea, will travel through the lens. What is it going to hit on the retina? What did we call them? The fovea centralis. That's where you have your most accurate vision, right? So it's directly dead in front of your field of vision. What's directly in front of you is what's going to hit that fovea, right? Most accurate vision. Now, we said that the cornea and the lens will bend light, right? So we're going to talk about that in just a little bit. I just want to keep that in your radar. Okay? All right. So we had the fovea, right? Kind of up here. Then we have this optic disc. Optic disc, does that have photoreceptors? No. That is your blind spot, right? And that is, again, the weak part of the sclera. If you have elevated pressure, this is where you're going to see the damage. We're just going to see it pushing out. Okay. So, need to talk a little bit about light. Light's really cool. It acts as a particle and it acts as a wave. Right? And we talked about this a little bit. When light rays encounter a new medium at an angle, they bend. Okay? So, right, we've got a lens. We're going to have our light coming in. It hits this lens right here. And that lens is going to bend it. That is called refraction, right? That is called refraction, that bending of light, okay? Now, I wear glasses, my husband wears contacts and glasses, right? When you are getting glasses, you need to know what lens you're getting and how much or how less it should bend light. So that's what we're going to talk about a little bit now, is bending a light and how we measure it, okay? You can measure the refracting power of a lens, and you do this by taking a measurement from the lens to where the light comes to a point, right? Where that light comes to a point, right? This is called the focal point, right? And for the distance from the lens to the focal point is called the focal length. Okay, focal length, right? So focal point, focal length. If we take 1 over the focal length, that gives us the refracting power of a lens. So 1 over focal length is the refracting power of a lens. Okay? Now, the larger the number, the greater the bending of light. So I want to provide an example for you of that, so you can kind of get that in your head about why that is, but we'll show you that in a minute. The larger the number, the greater the bending of the light, and the units we're going to use are called diopters. So 1 over the focal length, diopters, is the refracting power of a lens. Okay? So I've got a couple lenses up here, right? And you can see that each of the lenses has a different focal point. This lens really bends light, and this one doesn't bend it as much. You see that? Okay? So what I'm going to do is I'm going to give you focal length for each of those lenses and have you calculate the refracting power of each one. Okay? So, for this first one up here in A, actually I'm going to have you calculate B first because I think it'll be easier. I'll do B first and then I'm going to go back to A. B, we're going to use the focal length is 1 meter. 1 meter. So if we look at our equation, right, what is the refracting power of that lens? It's going to be 1 over the focal length. What's our focal length? Right, 1 diopters. Right? So it's going to be 1 diopter. Okay? Now, this one over here, A, what I'm going to do is it's shorter, right? So I'm going to have that one be 0.1 meters. 0.1 meters. Okay? So set up the equation. What's the equation for this going to be? 1 over 0.1. Okay? So what is 1 divided by 0.1? 10 diopters. Okay? Now what did we say about bigger numbers? The bigger the number, the more bending of light, right? And when you bend light more, that gives you a shorter focal length. You guys okay? Shorter focal length. Okay? Now, we've got one problem, right? And our problem is that not all lenses bend light to a point, right? Some lenses actually spread light out. Okay? So if I drew a lens that looks like this, sad, but nevertheless true, right? It would actually spread light out. Correct? Is this ever going to come to a point? No, right? So to calculate that type, right? Let me go back. These that cause the light rays to converge to a point are called convergence lenses. They converge the light, bring them together. Convergent lens, right? If you're going to spread them out, the rays are divergent, right? And you have a divergent lens. Now, we got a problem using our lovely equation, right? Because we can't come up with a focal length. So what we're going to do is we're going to draw an imaginary focal point in front of the lens. If we took these angles and carried them forward, could we come to a point? Yes. Right? So we now have an imaginary focal point, and we can calculate a focal length. So we're okay so far? Right. So if I said this is 0.1 meters, what is the refracting power of this lens? 10, right? But we have to be able to distinguish convergent from divergent lenses. Are we using an imaginary focal point, or are we using a real one? Right? If you have a divergent lens, it will be negative diopters. So negative 10 diopters. For that, what are we going to have to put in front of all of these? Positive, right? So have you ever looked at your prescription for glasses? It will have positive point something diopters or negative something diopters, right? And all of that, now you know what that is. Do you wear convergent lenses, or do you wear divergent lenses? Are you spreading the light before it comes into the eyes, or are you making the angles narrower before it comes into the eye? Right? Make sense, everybody? Okay. So what I want to do is I want to go ahead and stop here. Right? I have given you guys a handout. Has everyone gotten one? And this has eyes and ears on it. We haven't gotten to all the eyes yet. So what I want to do is have you start on number 6. Right? So you're going to do number 6, number 7, number 8, number 9, 10, and third page. Okay, so 6 through 10 and labeling of the ear. Okay, and then we'll go regroup and go through this. luck with it.